In this video, we will discuss about this CAPPSID functioning, how this CAPPSID uh, is able to perform its function that we will discuss in this video. In the previous video, we have discussed the application. One of the applications of this CAPPSID is to delivery of virus inside the cancer cells. So for this CAPPSID, they have selected the bacteria that is S. Tripimurium or Salmonella bacteria, and when it is inside the cells, this sp2 will be able to detect these intercellular parts and then trigger the translations of the viral circuit that is present inside the cells within the SCV. And once this is been activated or the parts has been translated, then it will cause the lysate of the SCV using the proteolytic proteins such as hemolysin E, and thereafter the viral parts will be extracted out and it will infect the host cells. So first part that they check is the translations of the viral proteins once they infected the host cells. So here they can see that at 0 hour signals are almost negligible for this PSSE M-cherry and this PTEC GFP. So they use two different promoters, SSE M-cherry and PTEC GFP. For GFP they can see some of the signals even at 0 hour, but for M-cherry they do not see any signals. And at 12 hours they can see the high signals for the GFP, or that is connected with this PTEC promoter, but PSSE they can see the signals only when it is been transfected. At 0 hour they do not see any signals for the M-cherry, indicating that this promoter should be good enough for their functions. And this is the quantifications that they have shown, is that for this normalized reporter value, we can see that at for this M-cherry by GFP, it increases with time. That can be seen here. And next I wanted to see is that whether their probe will be able to work once it is where their probe will be able to work uh, once it is in the cytosols of the host cells. So for that, first they check the viral replications using the GFP promoter, and these viral replications will only occur in the presence of the polymerase, and this results has been shown in the supplemental figures. And in that case, what they did is they inactivate the viral polymerase, and thereafter they can see the 52,000 folds decrease in the GFP signal, indicating that the viral polymerase is required for these viral translations. And in this case, that is the main figure. They have shown the results where what they did is they transduce this T7 polymerase, which is necessary for the translations of the viral particles or viral RNAs. So what they did is they infect this circuit of bacteria with this circuit into the cells, or we can say that is SM fish probe, where they can see the TAPI signals are there, and for the bacterial M-cherry signals, which has been shown here, which shows the translations of this T7 polymerase, which shows the translation of this T7 polymerase, which can be seen here. And this viral RNase SM fish, viral RNase SM fish is basically the red signals that they can see. These red signals that they can see once the viral particles are being translated. So that can be seen in this viral RNA SM fish, and we can see that bacterial M-cherry and viral RNAs almost are similar to each other, indicating that the formations of this or the translations of the viral RNAs is occurs within the SCVs. Next, they wanted to check whether the lysis, uh, the cells, SCVs are able to lyse after the translations occurs or not. So for that they use two different promoters or two different lytic proteins. It is HYLE and E. And here, when they don't use the lytic proteins, they can see the signals are more condensed. But when they use the lysis protein, they can see the signals are more spread out, indicating that the lysis occurs uh, correctly. Then they check their systems in different cancer cell lines, and they can see that in most of the cancer cell lines, they are able to see the percentage GFP signals. Since they have the GFP promoters, they are able to see the percentage GFP signals, and the most of the cell lines are infected to a moderate levels. That can be seen here in the blue color, and this one cell lines seems to be at very high infection rate of around 4.2. Next, what it is, they wanted to check their system in the actual cells, in vitro cells. So what it did is this: they check, they transduce this polymerase with PSSE promoter, and this polymerase on this lytic proteins also with PSSE promoters, and this once this promoter is able to form the polymerase, they are able to form this GFP and RBC. So here they are able to see the GFP signal, indicating that this T7 polymerase are formed, and after that this translations is also occurs correctly. And when they check these double-stranded RNAs, they are also able to see the signals of the red fluorescence because of the complete translations of the double-stranded viral RNAs, for which this red signals is being shown. So in this panel D case, let me clarify this. First, these PSSC promoters form this the T7 polymerase, which is been indicated by M cherry, and the final RBZ is the indicated of complete replications or complete translations of the viral RNAs. Here they do not show the GFP signals. So this is how they validate that their capsid is functioning properly and specifically. But right now they didn't mention the specificity that it can only target the cancer cells. We will hopefully researchers has covered that questions in the subsequent part of the research. We will discuss all other main figures in our subsequent videos. That's all for this video. So thank you for your time.